Hi everyone, my name is Katie Billé and I'm a master's student working in Dr. McTammany's aquatic ecology lab at Bucknell University. Today I'm really excited to share with you a project we've been working on that uses in-stream habitat additions as a way to increase recruitment of aquatic insects to streams and to potentially improve our restoration project's in-stream ecosystems. So to get started, when we think of stream ecosystems, we're thinking of the interaction between the physical habitat, the stream's hydrology, and the biota living within that environment. And each of these parameters is of equal importance when considering the overall structure and function of an ecosystem, whether that be under pristine or impaired conditions. And when restoration of an impaired stream is needed, current methods such as bank stabilization, level lip spreaders, tree plantings and riparian buffers, and cattle crossings or exclusions directly address the hydrology and surrounding physical habitat, but they don't necessarily address all of the needs of the biotic community. However, when we go to assess the progress of our restoration efforts, we look to the health of the creatures living within the stream to determine if our efforts were successful, even though the biotic community may not have been directly addressed during the planning and execution of the restoration itself. This is certainly the case when we talk about aquatic insects and other macroinvertebrates, which are a group of animals that are most often used to monitor the health of our streams. Now, aquatic insects like this stonefly have really complex life cycles that involve interactions with the aquatic and terrestrial environments. Many of these insects rely on the presence of in-stream habitat like riffles to successfully complete multiple life stages. For example, riffles are particularly important for recruitment of insects that exclusively oviposit or lay their eggs on microhabitat like rocks or organic material. Riffles are also home to diverse larval communities that often serve as a source of colonizers to proximal downstream habitat. This reliance on in-stream habitat to complete their life cycle makes certain insect taxa, like those who exclusively lay their eggs on rocks, particularly vulnerable to population bottlenecks in impaired systems, where in-stream habitat is typically altered, simplified, or even destroyed. And so the goal of the project that I'm going to talk to you about today was to investigate the extent to which in-stream habitat limits both recruitment and community diversity of aquatic insects, either due to a lack of suitable oviposition habitat or to the isolation of in-stream habitat patches, both of which are typical in degraded stream systems. To answer these questions, we chose a small unnamed tributary to Chillisquaque Creek as our study location. This stream is located at the Bucknell University Chillisquaque Creek Natural Area, which is a university-owned property in Montour County, Pennsylvania. As you can see from the picture on the left, this stream is characterized by a relatively uniform channel shape with substrata composed of soft clay and loose detritus. This made this stream particularly ideal for our project because it completely lacked any riffle or emergent habitat. This project was executed in two parts, both of which took place in the same stretch of our study stream. In part one of our study, we tested whether recruitment and community diversity of aquatic insects in our stream was limited by the availability of oviposition habitat, both at the habitat unit scale, which for example could include riffles, pools, or runs, and or at the microhabitat scale, such as different rock sizes within a riffle. To investigate this, in August of 2019, we built 9 identical riffles throughout our study stream, one of which is pictured on the left here. The bulk of each riffle was made from 1 to 3 inch rocks, which we used to make a 1 meter long riffle that spanned the width of the stream. 12 large rocks of equal surface area were then added to each riffle, and the small submerged rocks and large emergent rocks both provided potential habitat for insects to oviposit their eggs on. To test whether community diversity of our study stream was limited at the habitat unit scale, we left our stream for 2 months to be colonized by insects. We then used a server sampler to collect insects from our constructed riffles, as well as insects from the muddy areas that dominated our stream prior to the riffle additions. Back in the lab, we identified and counted the bugs from each of the habitat types. From our initial results, we found that our riffles did increase the diversity of aquatic insects in our stream as a whole. This graph shows the relative abundance of insect taxa in the riffle habitat and mud habitat. Between the two habitat types, there were a total of 35 different taxa in our stream 2 months after the start of our experiment. The following 10 taxa were found in both our riffles and mud habitats. And the mud habitat was home to 10 unique taxa, which comprised roughly 5% of the insects found in that habitat type. What's really exciting is that our constructed riffles were home to 15 unique taxa new to our stream and constituted roughly 30% of the insects found in that habitat type. Of those 15 unique taxa in our riffles, 3 genera currently have literature documenting that their oviposition behavior requires rocks as a substrate to attach their eggs to. The oviposition behavior of the remaining 12 taxa are unknown, and as a whole, specific documentation of oviposition behavior of most taxa is rare. Moving on to our second hypothesis, that community diversity in our study stream was limited at the microhabitat scale, we also sampled each of our 9 riffles for insect eggs. From each riffle, we looked at 3 large emergent rocks and 3 small submerged rocks. For each rock sampled, we documented either the presence or absence of insect eggs, the rock's location from the banks, the water velocity at that location, as well as the rock's dimensions, including surface area above water level for emergent rocks. Every egg mass we found was described, photographed, and a subsample was taken back to the lab with us to be reared for identification. As for our initial results, we found 26 egg masses total across all 9 riffles, 88% of which were found on large emergent rocks, and the remaining 12% were found on small submerged rocks. This suggesting that the preference or use of microhabitats varies among selectively ovipositing taxa. We also found that this degree of selectivity varied within taxa who selectively oviposit on rocks. For example, we found egg masses from the genus Hydropsyche and the family Chironomidae on both submerged and emergent rocks, but we only found egg masses from the genus Baetis on large emergent rocks. Moving on to the second part of this study, we also wanted to test whether isolation of in-stream riffles limits recruitment and community diversity of larvae in downstream habitats. To study the effects of habitat isolation, we were strategic in where we built the riffles that I just discussed in part 1. When we built our 9 riffles, we constructed them in sets of 3, where the sets varied by inner riffle distance. The first set of riffles were built 15 meters apart. The second set were built 10 meters apart. And the final 3 riffles were built 5 meters apart. The sets of riffles were separated by buffer regions, and we included an upstream control reach above all of the riffles in our study. Since aquatic insects are quite small and traveling via the drift in the water column can be difficult, especially over long distances, we expected to see higher densities of insects below our set of riffles that were only 5 meters apart when compared to the other two sets of riffles that were built 10 and 15 meters apart, as well as our upstream control. To test this, we used baskets of rocks to collect insects from below each set of riffles, below each individual riffle, and from our upstream control reach. We took our bugs back to the lab where they were counted and identified. And then for our initial results, this figure shows the average insect density from regions below each set of riffle, as well as our upstream control reach. We found that larval insect density in downstream habitat was in fact influenced by upstream habitat isolation, as insect density increased when riffles were built closer together, with the habitat below the set of riffles built only 5 meters apart having significantly higher insect density than our upstream control. To revisit our key conclusions from parts 1 and 2 thus far, we found that recruitment and community diversity in our stream was in fact limited by in-stream habitat used for oviposition. We also found that oviposition behavior and the degree of selectivity in oviposition behavior varies by taxa. And finally, that habitat patch isolation decreases the number of colonizers to downstream habitat. To wrap up this talk, I'd like to acknowledge that this is still a work in progress as we have additional samples and hypotheses to investigate, but I'm really excited about the results we've seen so far. I'd like to thank Bucknell University and the McKenna Foundation for supporting this project, my committee members for their continued support and advice, as well as the faculty and staff at Bucknell for helping me find all of the materials needed to build our riffles. And finally, I'd like to thank our 4 awesome undergrads, Belinda, Katie, Jordan, and Alex, all of who helped me sample and identify the bugs in this project. I'd also like to thank the organizers for putting on such a fantastic virtual river symposium. And finally, I'd also like to thank you if you've made it to this point in the talk, and I look forward to hopefully interacting and chatting with everyone throughout the rest of the symposium.